Okay, hello and welcome to my talk. My name is Adriano Martinelli. I'm from IBM Research in Zurich and ETH Zurich. And today I would like to present to you a tool that we developed called Athena to analyze tumor heterogeneity in spatial omics datasets. And as we already have seen in previous talks, we have these new technologies, the spatial omics technologies that enable us to analyze tissue samples while preserving the tumor architecture or the spatial architecture of the sample. And here you see now an example of a tumor that is depicted here with different cell types that are in the tumor, like the, sorry, is the audio good like that? If I move, it's fine, okay. So here you see a picture of a tumor and with its different cell types. So here in blue we have some different normal cell types in these bluish colors, along with some different tumor subclones in reddish colors, and then also some cell types from the tumor microenvironment, like immune cells or stromal cells. And all these different cell types are now interacting with each other in this tumor microenvironment. And they can actually do that in different ways. So one way, and quite popular, is how they kind of compete with each other in this microenvironment for nutrients or like oxygen, and then try to outcompete each other. And another idea described by the cooperation hypothesis is that those different cell types, and in particular the tumor subclones in this microenvironment, can now actually also cooperate with each other by sharing different factors, like different angiogenic factor or proteolytic factors, and together by this cooperation they can actually also share some of the different hallmarks of cancer that I depicted here from a recent publication. And as you can see, I try to highlight a few of these hallmarks that are corresponding or kind of linked to some of the spatial features that are present or that are linked kind of to the spatial organization of the tumor. And with Athena, we try to capture or to quantify a few of those hallmarks, as you will see later. Yes, so but first, I would like to quickly talk again about the spatial omics technologies. So over the last few years, these spatial omics technologies enabled us to actually quantify proteins, so dozens of proteins, or even thousands of transcripts in tissue samples. So while preserving their actual spatial location within the tissue, and this is in contrast to the standard single-cell sequencing or standard single-cell technologies, where you just have kind of the abundances of the different cell types in the end. And yeah, so the main difference between those two groups, the spatial transcriptomics and the spatial proteomics technologies, is on one hand in the number of molecular targets that they actually quantify, and then also, for example, in the resolution they do that. But yeah, all of those technologies, they actually produce some kind of images or an image-like output. And here I would like to quickly talk about one of those technologies, the imaging My Cytometry technology, to give you an impression of how the whole workflow is to produce the data. So here at the top left, you can see a tissue sample in orange or red. So this square that is then stained by a panel of antibodies that is labeled with isotopes. And then one can use a laser to actually ablate part of this or particles of this tissue sample. So you actually destroy the sample while doing that, and then subject the ablated particles to a CyTOF measurement. And then you get for each ablated particle, you get the protein abundance at this particular spot. And as an output, you will then receive these images you see there on the right-hand side that kind of visualize or you show the spatial distribution of these proteins within the sample. So in the end, you have a very high dimensional image. And in this image, like each layer of the image is their own protein measurement in this case. Yes, and oh, we don't see all the pictures. Okay, strange. Okay, we should see some raw data there. This would be the images that I just showed, and then some cell masks. So the cell masks would be kind of segmentations of those high-dimensional images. So as we have seen before, these images, they just show the spatial distribution of the proteins, right? And what you actually want to have is the protein expression levels of each single cell. And for that, you would need to create a segmentation of that image. And at the moment, Athena does not do that internally with the tool, but there are many good third-party tools that can do that. And then together with this, the raw data and the cell masks, you would be able to kind of compute the single cell expression values. Oh, sorry, the single cell expression values for each cell. That would be in this third picture that is missing. And then what we do with Athena, we convert those images into a graph representation, which is much lighter than the image itself. And it also allows you to kind of model the interactions within the tissue now. And here, as you can see now in this graph, we have these different nodes in the graph, and this would be the kind of segmentation that you used. So if you segmented your image into different cells, then each node in this graph would be a cell, and the different colors represent different cell types. And this graph representation is then the main input in our quantification module, where we characterize different aspects of this graph and also quantify different interactions that we see in this graph. And those quantifications you can then extract with our tool and then use for further downstream analysis tasks. So for the rest of the talk, I will use a breast cancer cohort data set, which was produced with this imaging mass cytometry technology that I presented earlier. And now I will just show some schematic visualizations of what we characterize in those graphs and how this then actually looks in those samples from this cohort. So here you can now see like the visualization of like 3 different proteins in a sample of this data set. And the color just kind of highlights the different intensities of these different proteins. And we can then use these different intensities to define different cell types within the tissue. So here we have some immune cell types, stromal cell types, or different also tumor cell types. And with the panel from this data set, so the antibody panel that was used actually also enabled the authors to produce a bit more granular cell types. So they, in the end, actually defined 27 different cell types that are shown here with different colors. And based on those images, we can now construct the graph, and Athena provides 3 different ways at the moment to construct the graph. So the first one is the radius graph, in which you connect each observation to all other, or each cell to all other cells within a given radius. And you could use that, for example, to kind of model paracrine signaling within the tissue. Another graph representation that we provide is the contact graph representation, where you connect each cell to the cells that are in contact with that cell. So you need to have physical contact that you connect the cells within this graph. And you could use that, for example, to model juxtacrine signaling. And then finally, we have the KNN graph representation, which is, it's just a popular graph topology or graph representation. Okay, and now the first kind of method that we implemented, or that you could use now to quantify different aspects of this graph, is the modularity. And here you see schematically 2 different tissue architectures. So one with a low modularity, where the different cell types are very intermixed with each other, and then one with a high modularity, in which you can actually segment the graph in different subgraphs that are very pure in terms of single phenotypes. And here you now see 2 examples from this breast cancer cohort data set, one with a very high modularity on the left-hand side, and one with a low modularity and a very intermixed topology on the right-hand side. Next, we also implemented several so-called information theoretic scores. So these are entropic scores, that tried to quantify the heterogeneity in the sample based on the number of cell types that are present in the that are in the sample, and also the abundance of the sample of the cell type. Sorry, and here you can see one with a low entropy and one with a high entropy. And so the highest entropy that you can achieve kind of in a sample is when all the cell types that are present are equally or uniformly distributed. And this then kind of, you can interpret this as the heterogeneity or the diversity in the sample is high, because like if you just sample a cell type by chance from this core, it's the most uncertain to predict which type it is. Because on the left-hand side, if one cell type is like much more present than other cell types, the probability to sample this particular cell type is much higher. And with that, you can then also analyze that here on these two example cores. On the right-hand side, you have one with a high entropy and one with a low entropy on the left-hand side. And in the histograms below now, you see the richness for both samples is approximately the same. So the richness is the number of cell types. But as you can see, the Shannon entropy between those two examples is quite different. And this is due to the fact that in the example on the left-hand side, one phenotype is it's like dominating all the other cell types in comparison to the one on the right-hand side. However, this kind of score actually is a score that is computed on the whole sample level. So you get one value per sample, and not really per single cell. So you don't quantify what's the heterogeneity or the heterogeneity of the environment that a single cell experiences, but what's the diversity in one sample. And to provide also kind of an estimate of how heterogeneous the environment of a single cell is, we adapted these entropic scores a bit to actually use now this graph structure to compute the cell type distributions based on the neighborhood of a cell within a graph. And here you can see now 3 different graph topologies from left to, so on the left-hand side, a very well-structured topology where a cell is surrounded by mostly other cells that are of the same cell type. So you could argue that the environment of this cell is very homogeneous. And therefore, if you look at the histogram at the bottom, the entropic values are skewed to very low values towards 0. And on the right-hand side, you have a very intermixed core where the different cell types that are around the single cell are very different, and therefore the entropic values or the distribution of the single cell entropic values is also skewed towards higher values. And again, this is visualized here with 3 different cores from this cohort. At the top, again, the cells are colored according to each phenotype. And then in the middle, we have in the middle panel, each cell is colored according to its entropic value. And in the end, at the bottom, you have the histograms of the single cell entropy values, where you can see that the core on the left-hand side, this clustered core, the distribution is highly skewed towards this zero entropic value. And on the right-hand side, it's much more skewed to higher values. Good. Another interesting aspect when you quantify or look at tumor samples and spatially in space, let's say, then this immune infiltration score, like the degree to which the tumor is infiltrated by immune cells, is of interest. And earlier publication by Karen in 2018 actually defined the so-called infiltration score, where they counted the number, or the infiltration score in the end is a ratio between the number of immune and tumor cell interactions, and this number is then normalized by the number of immune cell interactions. And you can now here see 3 different topologies, like a very compartmentalized tumor in A, where the immune cells and the tumor cells are highly separated. And this is in contrast to C, where the immune cells and the tumor cells are very intermixed. And in this original publication, this score was again defined on the whole sample level. So you looked at all the tumor immune interactions within a sample, and we adapted it again to kind of use the graph representation of the sample to compute these infiltration scores for each each cell in the sample. And again, when we visualize that for each single cell and then plot the histograms, we can now also see how you can distinguish between these three different tumor topologies based on the histograms. And finally, we also have some cell interaction scores where we actually try to quantify the interactions between different cell types. So how strongly two cell types are in contact with each other, or actually try to avoid each other. And you can then highlight or visualize these interaction scores with heat maps, where you can see in reddish colors are cell types that are attracted to each other, and in bluish colors cell types that try to avoid each other. And again, here the same three courses as for the whole presentation. Yeah, you see just how for the different tumor architectures, also visually these heat maps actually differ. And now I would like to quickly talk about the implementation of Athena. Yes, so at the core, so to store all these data sets, we introduced this Spatialomics data structure which is highly inspired by the N data structure that is used by GenCompBio, and you may recognize a few of the attributes like the ops attribute or the var attribute. But then we also have some very specifics or attributes that are very specific for spatial omics data sets, these images or masks attribute. And in this Spatialomics data structure, we just store all the data that is associated with this with a certain data set. And then we have our Athena module that has different submodules, like the plotting module, with which I produced all the graphs that you have seen here in this presentation, or the graph submodule, with which you can then actually compute or construct the graph representation based on the images. And these two modules kind of interact with each other, very similar to also to GenCompBio and then data. So you have a function in Athena, and you would provide the spatial omics instance to this function, which then extracts from the attributes all the necessary information to compute, for example, a graph representation. Yes, and yeah, with that, I'm at the end of my presentation. Thanks for your attention, and I'm open for questions.
All right, we have time for a couple questions. If you have questions, please come to the microphones in the middle.
Nice talk.
Hi.
Nice use of graph theory for biology. I had a question. So, you in Athena, you propose using a couple of different types of graphs, like the radius graph, the cells that are actually overlapping, and the nearest neighbor graph. And then you apply different metrics, and like one of them, for example, is the modularity metric, which kind of gives you an idea of heterogeneity as just like area of your graph. I was wondering if Athena provides some sort of like recommendation for which graph you should use for calculating those scores, and if you've done any sort of quantification of which graph is better for which method.
Okay. So the question is if Athena provides some recommendation for what graph type to use for which metric, and if I did some benchmarking for the different graph topologies. So we do not recommend to use a certain graph structure per se, but there are some types of scores like the infiltration for which you should use, for example, a radius graph to just the way how it computes the infiltration for each single cell. It's better if it has some more neighbors than just the ones that are in contact with each other. But per se, we don't suggest to use a certain score. And I think it also depends on the biological question you have, maybe what type you want to use, right? And in terms of benchmarking, I did not really benchmark the difference between the scores based on the different graph topologies yet.
I think that would be an interesting question.
Yeah, absolutely, I agree. Yeah.
What's the better way of like quantifying heterogeneity or other?
Yeah, absolutely. Yeah.
Well,
Thanks a lot.
Hi. So for a modularity score, did you consider only the graph topology, or did you also consider the cell type labels?
You also need to consider the cell type labels for the modularity, yes. So you have the cell type labels in the graph, and then the graph topology, and then with the modularity, you kind of compute like how easily you can kind of compartmentalize the graph into different subgraphs that are composed of the same cell type.
Oh, that makes sense. So for the cell-cell interaction score, did you only consider the spatial locations of the cells, or did you also look at the abundance of the ligand
Sorry, if I only use the cell type locations or the protein abundances.
Right. So when you calculate the cell interaction score, right?
Yes.
So you were looking into how likely two cells are interacting with each other.
Yeah.
So was that calculated only based on whether two cells are close to each other or in contact with each other, or did you also look at particular ligand and receptors activity levels?
So it actually does not, well, it depends on the distance that the cells are in the tissue, but it depends on mainly on the graph structure, because if you choose a graph structure in which you only connect the cell types or the cells that are in contact with each other, you will only those cells will then have an edge in the graph. So it depends on your graph topology, what kind of cells are connected within the tissue, and then only on that actually. So the phenotype that you assign to a cell and the graph topology. But not, I mean, the protein abundance is kind of reflected in the phenotyping that you do, right? You could also just binarize the data and say, okay, all cells that have a protein A level higher than X are positive and the rest is negative, and then compute the interactions between positive and negative cells based on a single protein expression level, more or less.
Okay, so that means when you were constructing the graph itself, you already considered the expression levels.
No, for constructing the graph, the expression levels are not considered.
So
it's just
is the expression level considered at any step? Like
in the phenotyping step only. Yeah, that's it. Okay.
So sadly, we're out of time.